Let me welcome you back to my course Aspects of Biochemical Engineering. Now, in the last lecture, you can remember I tried to discuss some numerical problems, and I analyzed try to analyze the batch and fed batch process. So we could just to find out that what time we can get that maximum cell mass concentration in a batch process, and also in the fed batch process we can find out that how much maximum cell mass we can produce and what is the time required for the fed batch operation. All these things we try to analyze in the last lecture. Now, this lecture is little bit different because we will be again discussing with the different numerical problems, but mostly we will be concentrating on the chemostat process. Now, chemostat now process is considered as a CSTR. CSTR is the continuous start tank reactor. As you know, when we use that in the biological system, we call it chemostat. Now, chemostat is a kind of process which can be very easy to operate, and not only that, productivity of the chemostat process is much high as compared to batch process. Only I told you the problem is the contamination we have in the during the long time operation. Otherwise, this is the process through which we can get the maximum amount of product formation. So, let us start with, because we will be taking the example of the different fermentation process. As you know, the Saccharomyces cerevisiae can be used both for Baker's yeast production as well as of ethanol production. Both the fermentation process we will be dealing with, and plus we want to discuss some other numerical problems. Now, first let us start with that the Baker's yeast fermentation process. Now, if you look at the Baker's yeast, the problem is one Baker's yeast industry produces 1 metric ton of compressed yeast. Now, what do you mean by compressed yeast? Compressed yeast means that, as you know, that the Baker's yeast industry, they usually produce two type of yeast. One is called compressed yeast, another is called active dry yeast. Compressed yeast contain more than 70% moisture, but in the active dry yeast contain about 6% moisture. So, active dry yeast whenever we produce, this is used for longer time of operation, and compressed yeast whenever we produce, that is for very short time, because it contains 70% of moisture. Since the moisture content is very high, so there is every possibility of contamination, because any bacterial cell can grow, because it contains we know that yeast contain about 50% of protein, and also it contains vitamin B. So, any bacteria can grow very easily in the yeast cell. So, this compressed is usually to be marketed under the refrigerated conditions. So, this problem deals on the basis of that. Now, that 1 metric ton compresses that and per day using the cane molasses as the raw materials in a chemostat. Compressed yeast contain about 70% weight by weight moisture. Mu max, Ks and Y x by S value are given 0.5 hour inverse to 2 gram per liter of Ks, and Y x by S is 0.5 gram per liter respectively. Cane molasses contains about 45% weight by weight sucrose. Initial substrate concentration of the fermentation broth is 200 grams per liter. Compute the followings. What we shall have to compute? We shall have to find out what is the minimum doubling time of the cells, the total amount of cane molasses required, volume of the fermenters, and maximum cell mass productivity. So, this is, I consider this is a very important problem as per the biochemical industry is concerned, particularly Baker's yeast fermentation is concerned. And in this particular connection, I want to tell you that if you look at the most of the biochemical industry or industry that we have in India, that most industry they usually operated in a batch mode, but as soon as the World Trade Organization treaty was signed, then we have open trade in the different country. Then all the industry they switch over to the they convert batch to the continuous process. The beauty of the continuous process is that your percentage labor consumption is reduced to 10%, and your amount of product formation increases more about 10-fold. So, there is a huge amount of productivity increases. So, this problem is very important. So, when we start with 1 metric tons of compressed dry yeast per day, what do you mean by that? 1 metric ton means 1000 kg compressed yeast per day. Now, it contains about 70% moisture. So, how much total solid will be there? 30%. So, naturally 1000 into 0.3, that will be 30 kg dry yeast to be produced per day. Am I right? Now, first we shall have to find out that what I shall have to find minimum doubling time. Am I right? So, what is the minimum doubling time? We know that we have already shown you that ln x by x0 equal to mu into T. Am I right? Doubling time means double the cell mass. So, this is the doubling time. So, x0 will cancel. So, ln2 ln2 by mu is the T doubling time. Now, when this doubling time will be minimum? When mu is mu max, then it will be minimum. This is exactly what we have written here. D doubling time equals to ln2 by mu max, and ln2 is 0.693 and the mu max is 0.5. So, it is coming around 1.386 hour. That is the minimum doubling time that is required for the cells. Now, next problem is that the total amount of cane molasses required, that we shall have to find out. Am I right? So, the question comes how we can find out. Now, in this problem, that amount of cane molasses required per day, how we can find out. So, 300 we shall have to produce how much? 300 kg of yeast, that dry yeast. Am I right? So, and yield coefficient is 0.5. So, if you divide by that, you will find how much substrate is required, sugar is required. This is 600 kg. Now, let us assume the sugar conversion efficiency is 95%, because this is the theoretical value. Am I right? So, may not be the all sugar will be converted into the cell mass. So, let us assume that 95% is the conversion efficiency. Then actual amount of sugar requirement will be 631.5 kg. Now, cane molasses contain about 45% of weight by weight of sugar. So, if you divide by 0.45, then you will get 1.4 metric tons of the cane molasses. This is cane molasses that we required for this. So, second part of the problem can be solved very easily. Now, third problem that we have, that we shall have to find out the volume of the reactor. Now, how we can find out the volume of the CSTR? I told you can remember in CSTR, first we shall have to find out tau CSTR. What is the tau CSTR? Tau CSTR equal to S0 minus S divided by minus RS. Am I right? Now, once you know that, then this is equal to this is equal to this is equal to V by F. Now, if you know the flow rate, that volumetric flow rate, then you can find out the volume of the reactor. So, this is how you can find out the volume of the reactor. Now, here exactly that is written here, that this equal to tau CSTR equal to S0 minus S by minus RS. Now, minus RS, how we can write? Minus RS, we can write minus RS is equal to what? Minus ds by dt. Am I right? Now, this is equal to minus ds by dx by dt. Now, this is equal to dx by ds 1 by. So, this is 1 by yx by s into this is dx by dt is equal to what? This is equal to mu into x. This is exactly what we have written here. Okay. Now, from the Monod kinetics, we know mu equal to mu max S Ks plus S, and x is like this. Now, what is the question come, what is the maximum cell productivity? Maximum cell productivity means that we know there is a situation called D max. What is D max? D max is the dilution rate when you will get the maximum rate of cell mass production. Am I right? Now, so we shall have to first find out the D max, then we shall have to find out at D max what is the cell mass concentration. So, D into X, if you D multiplied with X, then you will get the maximum cell mass productivity. This is exactly what we are trying to do here. So, first we shall find out the D max value, and we find this is 0.45 hour inverse, and then corresponding S value we find out this is 18 point, S value is Ks into D max, mu max minus D max. So, we can find out this is about 18 grams per liter that we have. Then if you come to this equation, then we find that in the sterile feed, when you have sterile feed x0 equal to 0, and then by substituting this, the initial substrate concentration was 200 grams per liter, and final substrate concentration was that steady state substrate concentration 18 grams per liter. So, what is the x value will be y x by s into 200 minus 18, this is 91 grams per liter. Now, what is the RS value that we have written? This is the equation. Now, in this equation we can put that value of y x by s, mu max, then s, then ks, s and x, we can, all the values is available with us, and we find out this is 81.9 grams per liter per hour. Now, tau CSTR we have find this is S0 minus S by minus R is equal to V by F. Now, what is S0 value is 200 grams per liter, S is 18 grams per liter, and this rate of substrate degradation is 81.9. So, this will give the 2.2 hours. Now, we will have to find out, you see that if you look at the previous equation, this is equal to V by F. So, we shall have to, if we want to find out the volume of the reactor, we shall have to find out the flow rate. So, how you can find out the flow rate? Flow rate can be find out if what is the substrate required, that we have already calculated the 631 kg per day, and what is the initial substrate concentration is 200 grams per liter. Now, 200 grams per liter is equal to 200 kg per cubic meter. So, this will be converted into this, we can write in this form 3.1, this is cubic meter per day, and this we can convert in terms of the hour by divided by 24, then 1.131 cubic meter per hour. Now, we have find out that total time required for the CSTR is 2.2 hours. So, you multiply this flow rate, you will get the volume. So, this is the volume of the reactor we can calculate. Then at the same time I told you that how you can find out the maximum D into X. Now, what is the D value, D max value we calculate? D max value we calculate 0.45. What is the X? Let me see that. Now, maximum cell productivity we shall have to find out. Then X value we shall have to find out. That I think this X value that we shall have to determine, and the X value we find is about 91 grams per liter. So, so we can this unit is hour inverse. So, it will come. So, it will come around 41 gram per liter per hour. So, this is how we can solve this equation. Now, next problem also very interesting, that Pseudomonas species. Pseudomonas doesn't require any kind of introduction, because Pseudomonas is the first organism, genetically modified organism that was patented in the USA. And one of the Indian scientists you might be knowing, Ananda Mohan Chakraborty, he got the patent. And this Pseudomonas is very much famous for, is considered as the oil eating bacteria, because lot of oil spillage we have in the ocean, and this bacteria can remove that oil from the sea. So this problem deals with that Pseudomonas species, the minimum doubling time is being grown on acetate, and in a chemostat operation the monod, and these are the different value. Very simple things. What we want to know that what will be the value of x and x, s and x at the dilution rate d equal to half Dmax. So we can first we can easily find out what is the Dmax value. What is the Dmax value? This will be equal to mu max 1 minus root over Ks by Ks plus S0. Am I right? So we can easily find out the value of beta. So once we know that, we can find out the value of S. S is what? Ks into D max minus mu max minus D max. Am I right? So we can find out S value, and once you know the x value, we can find out the x value. X value is what? Y x by S into S0 minus S. So we can find out that the x and s value at Dmax, when Dmax is equal, d equal to half of Dmax, then you can find out also the value of s and x. And once you know that, then we can find out what is the value of 0.8 Dmax. Then you can find out the cell mass productivity. What is the cell mass productivity? D into x. And what is the cell washout? Cell D washout will be at washout there is no cell present on the reactor, then S will be S0, then the equation will be mu max S0 Ks plus S0. So I hope it is very simple problem. I hope everybody can understand it. So what we have written here, the minimum doubling time is can be written as ln2 by mu max, and then we can find out that what is the mu max value is about this, and once we know the mu max value, we can find out the Dmax value. Once we have Dmax value, we can find out the D value equal to half Dmax. This is the 0.2, this is our inverse, am I right? And then S value we can find out Ks D mu max minus this D is nothing but half Dmax. So you know that we can put this value, we can find out x equal to this, and x equal to this when we consider the sterile feed. If you have the sterile feed, then what will happen? That x0 equal to 0. Am I right? And then we can have this equation, we can put this is 70 grams per liter. Now when D equal to 0.8 Dmax, then we can this D value will be 0.188 our inverse. Now y x by s, y x will be x equal to what? Y x by s, S0 minus s. So we can easily find out the respective value of steady state substrate concentration and steady state cell mass concentration, we can find out. Now what is the productivity? Productivity is nothing but x into d. So we know the value of x, we know the value of d, so you multiply that, we will get the 3.7 gram per liter per hour. Now D washout, I told you that the initial substrate concentration is 38 grams per liter. If you put this value, then we will get the D washout value. So this problem is the typical problem. I hope everybody can understand, can solve by yourself. Now next problem is a very interesting problem, that is how we can analyze the cascade type of reactor. Cascade means that multiple CSTR, that one after another, if you do that. Because why we are interested? Because the reason is that when we operate a single chemostat, then you know that our problem is a little bit simplified, because this is single chemostat, so this is coming and going. Then we have S value, we know S value equal to what? Ks D mu max minus D, am I right? And x value also you can easily find out x0 plus y x by s, S0 minus s. So you can easily find out. But what will happen when this is connected with another CSTR? Suppose suppose that suppose this is the CST here, this is one reactor, again it is connected with another reactor. So, how you can analyze this? Now, this only the difference is that when we analyze the one reactor, we assume x0 equal to 0, am I right? But when you go for the second reactor, this is first and second, here x is not equal to 0, is not equal to 0. So, what you have to do? You have to write the cell mass balance in this equation, and then and only then you can solve this equation. This problem deals with this. I hope you will understand that. Let us see how we have done that. Now, the problem is that consider an organism which follow the Monod equation, and mu max value this, and Ks is 2 gram per liter. In a continuously perfect mixed vessel at steady state, no cell death, S0 equal to 50 grams per liter, Yx by S is 1. When dilution rate D will give the maximum total rate of cell mass production. So, what dilution rate D? So, that equation already we know that D max equal to mu max 1 minus root over Ks plus S0. We put the different value, we can find out the D max. So, D max we do not have any problem, because we can solve it very easily. Now, question comes is the second part. For the same value of D, using tanks of the same size in series, how many vessels will be required to reduce the substrate concentration to 1 gram per liter? That means, what it says that you have multiple number of tanks, am I right? So, this is connected like this. All are continuously start tank reactor like this. So, this is number of reactors. So, here we have 50 grams per liter. So, problem is that this is 1, 2, 3, 4, 5, 6. Now, how many that reactor will be required to reduce the substrate concentration to 1 gram per liter? This is the problem that we have. Now, let us see how we can solve this problem. Now, first we say what we shall have to find out, that we shall have to find out that what is the, this is like this, schematically this process is like this. This is the initially F is the flow rate, F is the flow rate, then x0 is the initial. So, this we can assume to be 0 in the first reactor, am I right? x0 equal to 0. But then we can have this equation, we can find out the value of x1, we can find out the value of S1. So, S1 value is, we can write d Ks minus divided by mu max minus d. So, we can find out 8.2 grams per liter. Now, we can find out x1 value Yx by S equal to, this is 41.8 grams per liter. The first reactor we do not have any problem. The second reactor we shall have to find out that what is the value of x, and then we shall we are interested to find out the value of S2. Because then if it is more than 1 gram per liter, we shall have to analyze again for the third reactor. So, let us see that in the second reactor what should be the substrate concentration. For doing so, what we can do, we can write the substrate balance, and if we write the substrate balance, this equation will come. This is equal to let me show you this, how this equation has come. Suppose what I was telling, this is first reactor, this is second reactor, am I right? So, this is like this. Now, here you have x0, S0, this is S1 and this is S2, am I right? And this is x0 equal to 0, and this is x1, this is x2. Now, if you want to do here the substrate balance under steady state condition, what we can write? What is the rate of input of substrate? This is F is the flow rate, F is the flow rate here, F is the flow rate, all the flow rate is F. So, F into S1 is the what is the generation of the substrate will be 0. What is the rate of output? This is equal to F into S0, S2, am I right? And what is the rate of substrate consumption? it is ds by dt into x, sorry, this is ds by dt into V, this will be V, and rate of accumulation that will be equal to 0. Am I right? So, this is the equation that if you divide by V both the sides, this is V, this is V, this is V, then what we can write, this is equal to d into S1 minus S2. What we can write, this is equal to ds by dx by dt, that we have shown you before. This is equal to yx by s, and this is mu dx by dt equal to mu into x, and then I can write this is 1 yx by s, this is mu, this will be x2 second reactor, am I right? Then this will be mu max into S2 plus Ks plus S2, am I right? So, this is into x2, and what is the value of x2? X2 will be equal to, this is yx by s x1 plus yx by s into S1 minus S2. So, we can put this value here, and then there will be a quadratic equation, only S2 will be unknown, single equation, single unknown, we can easily solve it. So, in this problem, this is exactly what we have written here, and then we can put this equation, this is the equation has been built up, and finally, we can find out the substrate concentration. Now, this is coming about 0.293 grams per liter, that means this is less than 1 gram per liter. So, our answer is 2 reactor will be enough to reduce the substrate concentration to 1 gram per liter. The last problem that we have with the distillery industry, one distillery industry is producing 100 cubic meter of rectified spirit containing 90% volume by volume of ethanol in a chemostat from cane molasses, containing 50% weight by weight sugar using Saccharomyces cerevisiae. The characteristics of the yeast is given here. You have to find out the volume of the bioreactor and amount of cane molasses is required. The almost similar to the first problem, only here the product is different, here product is ethanol. So, here x0 we can, if you consider sterile media, x0 equal to 0, then steady state cell mass concentration will be x equal to x0 y x by s s0 minus s, Dmax equal to this value, am I right? And ethanol is considered as a growth associated product. So, we assume if we operate in the Dmax, we will get the maximum amount of ethanol production. So, this S value is this, and this is the cell mass concentration. Now, 100 cubic meter of spirit we shall have to produce, which contain 90% of ethanol. So, this will be 90 cubic meter of ethanol. 90 cubic meter ethanol is 90,000 liters per day. The ethanol density is 780 grams per liter. Then we find out what is the total amount of ethanol is to be produced. This is the total amount ethanol is to be produced per day, and if we know y p by s, that is the yield of this of your how much product ethanol is produced per gram of substrate, then we can find out how much sugar is required. Now, what is the flow rate? Flow rate equal to amount of substrate required divided by initial substrate concentration. So, we can easily find out this is the flow rate, and once we know the flow rate, then you come to tau CSTR equal to S0 minus S minus RS, RS equal to this RS value we can easily calculate it. We find out this is the RS value, similar to the first problem that I explained. I am not explaining again here, and we find out the value of tau CSTR is equal to V by S0 minus S by minus RS equal to V by F. So, we know this value, this is 300 minus 23 by that RS is 12.74, and this is equal to 21.74 hours. So, V equal to what will be this is the flow rate that we have, this is the hour. So, the volume of the reactor will be 423.93 cubic meter. Now, substrate required per day. So, how you can calculate? We can calculate that how much substrate we required per day. We can easily calculate. This is already we have found out this much of substrate required. Now, it contains about 50% of sugar. Cane molasses contain 50%. So, you multiply it by 2. We multiply it by 2. We will get the amount of cane molasses required for this distillery industry. So, what I conclude here that chemostat process we can analyze very easily, and it is a very advanced process, and we try to explain that in a chemostat how you can get the maximum cell mass productivity, how you can get the maximum ethanol productivity, how you can find out had different dilution rate, how we can find out the substrate concentration and cell mass concentration. Also, I try to analyze the cascade reactor. If you use that multiple chemostat, how you can analyze the multiple chemostat process. Thank you very much.